My name is Ritika Balyan. I am a student of Chemistry Honours 3rd year, Kirodimal College. Topic for my presentation is Nomenclature of Nucleosides and Nucleotides. Deoxyribonucleic acid DNA and ribonucleic acid RNA are essential biological macromolecules that carry genetic information. They are polymers made up of repeating monomer units called nucleotides. Understanding the systematic nomenclature of these units is fundamental to biochemistry and chemistry. Nucleotides are constructed from three main parts. Nitrogenous base. It is a heterocyclic ring structure containing nitrogen atoms, purines or pyrimidines. Pentose sugar and phosphate group. 1, 2 or 3 phosphate moieties linked via ester and phosphoanhydride bonds. Whereas nucleoside only consists of a nitrogenous base and pentose sugar. It does not contain a phosphate group. This is what differs nucleotide from nucleoside. So nitrogenous base are planar, aromatic and relatively water insoluble. They are derived from purines, which are adenine A, guanine G. Pyrimidines are single ring structures, cytosine C, uracil U, thymine T. Uracil is typically found only in RNA, whereas thymine typically found in DNA. These are the structures of adenine, guanine, thymine, cytosine and uracil. Now ribose and deoxyribose. The pentose sugar dictates whether the nucleotide is part of RNA or DNA. If it is a part of DNA, then it has deoxyribose, and if it is a part of RNA, then it has ribose sugar. 2D-deoxyribose sugar is found in DNA and lacks the oxygen atom at the 2 carbon position, whereas the ribose is found in RNA and has a hydroxyl OH group at the 2' carbon position as shown in the figures. Nucleoside formation. A nucleoside is the combination of nitrogenous base and a pentose sugar. The bond connecting them is an N-glycosidic bond as shown in the image, and this bond forms between N1 of pyrimidine or N9 of a purine and C1 of the pentose sugar. The formation involves the removal of a water molecule. These are the structures of some nucleotides and nucleosides. Now coming to nucleoside nomenclature. The systematic name involves changing the suffix of the base name. Purines, that are A and G, adenine and guanine. The suffix changes from ine to ozine, and pyrimidines, that is C, T, U, thymine, uracil and cytosine. The suffix changes from ine to id. The prefix deoxy is added for DNA components because DNA components have deoxyribose sugar. For example, as shown in the figure, the base adenine is known as adenosine if it is a ribonucleoside, and it is known as deoxyadenosine if it is a deoxyribonucleoside. Same for cytosine. If it is a ribonucleoside, it is cytidine, and if it is deoxyribonucleoside, it is deoxycytidine. Same here it is given for guanine, uracil, hypoxanthine and thymine. Now coming to nucleotide formation. A nucleotide is a phosphorylated nucleoside. The phosphate group is typically attached to the C5 carbon of the sugar via a phosphate ester bond. Multiple phosphates can be linked to the first phosphate via phosphoanhydride bonds, which are energy-rich bonds. The addition of the first phosphate makes a molecule an acid, example adenylic acid. As it can be shown in the figure, a nitrogenous base plus a sugar is a nucleoside, whereas if it consists of a phosphate group, it becomes a nucleotide. Now the nomenclature of nucleotide. Naming depends on the number and position of the phosphate groups. Append monophosphate, diphosphate or triphosphate is added to the corresponding nucleoside name. We specify the carbon position, that is usually 5. Abbreviations: the first letter indicates the base, the last indicates the number of phosphate. A lowercase d prefix indicates deoxyribose, which is in this case of a DNA. As it is shown, adenosine 5 monophosphate is abbreviated as AMP, where adenosine is the name of the base and MP is monophosphate, which can be changed to DP if it is triphosphate, DP if it is diphosphate, and a small d prefix is added to the name if it is formed through deoxyribose sugar. Now alternative naming and occurrence. Violate suffix. Nucleoside monophosphate can also be named using the suffix violate. Example: adenylate, guanylate, cytidylate, uridylate. Acronyms. Simple capital letters A, G, C, T, U often represent the base within a sequence context, but in chemical terms they usually represent 3,5 monophosphate form. Now the summary of nomenclature. The systematic ring of nucleoside and nucleosides rely on identifying the specific base, the sugar type, which could be ribose or deoxyribose, and the number position of the attached phosphate group. Here are some examples of DNA and RNA names with components. Thank you everyone.